Hello, we'll be discussing three important topics in microbiology, which is very much important for your upcoming exams. That is staining, culture media, and sterilization. We'll start with the history. One important point which I felt which has been asked in previous exams, your entrance exam, is who is the father of modern microbiology? So father of modern microbiology is your Louis Pasteur. Okay. So he is the father of modern microbiology. He has tremendous contribution in microbiology. He has given the principle of fermentation. He has given the sterilization technique like your autoclave, hot air oven, and steam sterilization. The milk which we use in our routine purpose is pasteurized milk. That also pasteurization technique was given by him. The previous year, which has been asked in a recent exam question, who has given the germ theory of disease? So germ theory of disease is given by father of modern microbiology, which is Louis Pasteur. He has given the concept of liquid culture media. Now coming to staining, there has been question on staining technique. The gram stain, Albert stain, acid fast staining has been asked in previous entrance exams in AIMS PGI. They have asked what are the steps in staining. You should be knowing the steps of gram staining. You know the gram staining is most important staining which we do in microbiology to differentiate your gram positive from gram negative organism. So we have primary stain, which is your gentian violet, crystal violet, and methyl violet. So all three are your primary stain. Okay. Then you have gram iodine, which acts as a mordant, which fixes the dye. Okay. So you have given the sample or smear primary stain, which is your crystal violet or methyl violet. Then you are giving the fixative of that stain, that is your mordant or gram iodine. Then you are decolorizing it with the acetone. Okay. Then you're giving the counter stain, dilute carbonation, and you should be knowing how much time duration it takes. So your primary stain takes 1 minute. Gram iodine fixative takes 1 minute. Your decolorization takes few seconds, 2 to 3 seconds. Your counter stain, that can be dilute carbonation or safranin, which takes around 30 seconds. So in your previous entrance exam, it was asked the order of gram staining. So please remember, you're using crystal violet, methyl violet, gentian violet, all three can be used. Then you have gram iodine, then you have decolorizer acetone alcohol, then dilute carbonation or safranin. So this is the order of gram staining. Now coming to Albert stain. You must be knowing Albert stain, which we used for Corynebacterium diphtheriae. So this is your Corynebacterium diphtheriae Albert stain picture where you are seeing green bacilli and bluish black metachromatic granule, which we see in Albert stain, which we do for Corynebacterium diphtheriae, which causes diphtheria. Okay. So it has been asked what are the components of Albert stain. So Albert stain has two components, Albert A and Albert B. Okay. Albert A has toluidine blue, it has malachite cream, it has glacial acetic acid. Apart from it has 95% alcohol, which we finally all these dye we dissolve in alcohol and add distilled water and acetic acid. So we have three dyes, toluidine blue, which will give blue color, you know, malachite green color, which are dissolvent or solvent. We have glacial acetic acid, we have alcohol and distilled water. Albert B, we have iodine and potassium iodide. So it has been asked the constituent of Albert stain. So please remember, toluidine blue is there, malachite green is there, glacial acetic acid is there, 95% ethanol or ethyl alcohol is there, which we finally add in the distilled water. Then you have iodine and potassium iodide. Coming to next, acid fast staining. Acid fast staining is a staining technique which we use for diagnosis of Mycobacterium tuberculosis. Okay. And acid fast staining, we have Ziehl-Neelsen staining, we have Kinyon staining. Okay, Zeal-Nielsen staining and quinone staining are type of acid-fast staining. Zeal-Nielsen, we use heat. Quinone, we don't use heat. So we say cold method of acid-fast staining. The steps of acid-fast staining has been asked in your previous AIMS and PGI. That is what are the steps? So we use primary stain, which is dilute carbon fusion. Then you have decolorizer, which is 20% sulfuric acid, and methylene blue or lawless methylene blue, which is counter stain. So you have strong carbon fusion. Please remember, it's not dilute carbon fusion. It's strong carbon fusion, 20% sulfuric acid, and methylene blue. So this is the order which we use for acid-fast staining. We have modification in acid-fast staining. We modify it. How we modify it? We change the percentage of sulfuric acid. Okay, here we are using 20. When we use 5%, we use for Mycobacterium leprae. When we use 0.5%, which we use for oocyst of parasite, which all parasite, coccidian parasite, Cyclospora, Isospora, and Cryptosporidium, right? And 1% sulfuric acid we use for nocardia. So it has been asked 0.5%, 0.5% sulfuric acid is used in which all diagnosis of which all parasitic disease. So you can say oocyst of Cyclospora, Isospora, and Cryptosporidium. Now coming to next important topic, culture media. So microbiology is based on culture media, right? So we have how many types of culture media? We'll start with types of culture media. First one is your simple or basal media, which is nutrient agar. You can see the picture of this because you get the picture-based, case-based questions. So please remember, this is your nutrient agar, which supports the growth of non-fastidious organism. Okay, fastidious, which requires extra nutrition, non-fastidious, which can grow on the simple media. Okay, so what is there? Nutrient broth is there and 2% agar is there. So the percentage of agar has been asked previously. How much percentage of agar? 2% of agar. Okay, when you want to make it a semi-solid, you use 0.2 to 0.5% agar. Please remember, you are using 2% agar in nutrient agar, which is a solid media. When you want to make it semi-solid, you have to decrease the percentage of agar to 0.2 to 0.5%. Where we use this nutrient agar? We use for reading the colony morphology, pigment production of the bacteria. We know few of the bacteria produces pigment like your Pseudomonas, Staphylococcus aureus. So for their pigment production, we need nutrient agar. Then if it's semi-solid, we demonstrate the motility of the bacteria. Now we have enriched media. Moving to next media, enriched media, which is basal media, that is your nutrient agar, where we add blood, serum or egg. Okay, anything which provide special nutrition. So it can be blood, it can be serum, it can be egg. So this is used for non-fastidious organism, which requires extra nutrition, special nutrition like Neisseria, Streptococcus, Haemophilus. Okay. So examples of enriched media is your blood agar, chocolate agar. Blood agar, 5 to 10% of sheep blood. We add nutrient agar at 45 degrees centigrade to demonstrate hemolytic property. Chocolate agar, we use 5 to 10% of sheep blood and we add nutrient agar at 70 degrees centigrade. So it gets hemolyzed and nutrition from RBC, factors from RBC gets released. Chocolate agar is ideal media for the growth of Haemophilus influenzae. Now, Loeffler serum slope. So this is your blood agar picture. This is your blood agar picture. This is your chocolate agar picture. Now this is your Loeffler serum slope. Slope is slanting position, right? So this is your Loeffler serum slope, which we use for the culture of Corynebacterium diphtheria. Okay, so that is your Loeffler serum slope. That is your third enriched media. Now coming to enrichment media. So we read about basal media, we read about enriched media, that is your blood agar, chocolate agar, Loeffler serum slope. Now we are moving to enrichment media. Enrichment media is liquid media. Your enriched media was solid media. Enrichment media is your liquid media, which has some inhibitory substance which allow growth of some organism and inhibit some other organism, right? So when you want to isolate the pathogen from normal flora, you use enrichment media. So you must be knowing, you have seen plenty of question on enrichment media. Even your CET this year, CET has been asked for Shigella. So you use tetrathionate broth for Salmonella typhi, you use selenite F broth for Shigella, you use gram-negative broth also for Shigella, you use alkaline peptone water for the Vibrio cholera. So these are the list of your enrichment media. Enrichment media is liquid media. So tetrathionate, Salmonella typhi, selenite F, Shigella, gram-negative broth, Shigella, alkaline peptone water, Vibrio. Now coming to selective media, the name indicates, right? It selects something, it inhibits something, okay? So they will have some inhibitory substance which will allow only pathogen to grow, normal flora to get inhibited. So you have algae media, you have TCBS, you have deoxycholate citrate agar, XLD, that is xylose lysine deoxycholate, you have PTA, potassium tellurite agar. Now coming to Lowenstein-Jensen media is used for, this is the picture of your Lowenstein-Jensen media used for Mycobacterium tuberculosis. Then you have TCBS. This is the TCBS, which is showing yellow colony of Vibrio cholera. So TCBS is for Vibrio species. Now you have DCA media. DCA, this is your DCA media, deoxycholate citrate agar, which showing growth of Salmonella, black color colony, that is your Salmonella. Okay. Then you have XLD. There also you see black color colony of Salmonella. So XLD and DCA can be used for Salmonella and Shigella both. This is your potassium tellurite agar, the last picture which we are seeing, potassium tellurite agar, which is used selective media for Corynebacterium diphtheriae, where tellurite is reduced to metallic tellurium and it gives black color. Now coming to differential media, name indicates differentiate between two, right? So differentiate between two group of bacteria which has some indicator. These differential media will have some indicator in this which changes color depending on the bacteria. So McConkey agar is your example for differential media. You can see this side is your non-lactose fermenter and this side is your lactose fermenter. Lactose fermenter gives pink color colony, non-lactose fermenter gives colorless colony. Your McConkey agar has peptone, it has lactose, it has indicator which is neutral red, okay? And it has a taurokelate. So this neutral red is the indicator in your McConkey agar, okay? Apart from McConkey agar, what is the differential media which we use is CLED media, cysteine, lactose, electrolyte deficient media. That also differentiate between LF and LF and we use for urine specimen. Now coming to transport media. All the media name is based on what is their use, yeah, right? The transport media will use for transportation of specimen. When you know that transportation is delayed, okay, or you are transporting some delicate organism like Streptococcus neisseria, you need transport media to maintain, it maintains the viability but does not allow bacteria to multiply. So you have different transport media for different bacteria. So for Vibrio cholera, we have VR, Venkatraman Ramakrishnan medium. We have Carrie-Blair medium. For Shigella, Salmonella, we have buffer glycerol saline and Carrie-Blair. Okay. For Neisseria, we have Ames media and Stewart medium. Anaerobic culture media, that was asked this year. Anaerobic culture media is Robertson cooked meat media, right? Which we use for, this is your RCM, which we say Robertson cooked meat media, which has a meat particle and it is used for anaerobes like Clostridium. Now moving to third topic. Okay, so in culture media, we saw the types of media, which is your simple media, enrichment media, an example for enrichment media, very important, okay. Selective media we saw, then we saw differential media, now transport media, anaerobic culture media. Now moving to sterilization and disinfection, right? Sterilization is very important topic. So what are the agents of sterilization? Okay, we have physical method, we have chemical method. Okay. So in physical method, you have autoclave, hot air oven, filtration, radiation, microwave, that all is incineration. Those are physical method of sterilization. You have chemical method of sterilization. Sterilization, when you destroy all pathogen, including the spore form, that is called sterilization. Chemical sterilization, you have ethylene oxide sterilizer, which we say ETO, commonly said ETO. Then you have plasma sterilizer. Okay, so this was sterilization. We destroy pathogen and spore both. Then you have disinfect. You have high level disinfectant, you have intermediate level disinfectant and low level. Please remember which all comes under high level disinfectant. You have aldehyde, glutaraldehyde, orthoptaldehyde, formaldehyde, that comes under high level disinfectant. Peracetic acid, hydrogen peroxide. Then you have intermediate level. Intermediate level, you have physical, pasteurization, boiling, and all comes under intermediate level of sterilization. Chemical, you have ethyl alcohol, isopropyl alcohol, phenols, halogens comes under intermediate chemical method. Low level disinfectant, we have quaternary ammonium compound and chlorhexidine. Spalding classification for devices like critical device, semi-critical device, non-critical device. Critical device is your implant, scalpel, surgical instrument, where we use sterilization or disinfectant. Semi-critical, which is your endoscope, laryngoscope, endotracheal tubes, we use high-level disinfectant here, okay? And non-critical device like your thermometer, stethoscope, bedpans, their thermometers, we use intermediate level disinfectant. Stethoscope, we use low level disinfectant. Now coming to autoclave. Autoclave, we know the temperature is 121 degrees centigrade for 15 minutes, 15 pound pressure per square inch. Okay. How it acts? By coagulation mechanism. Okay. It coagulates, denatures the protein and enzyme. So these are the picture of your autoclave. You have horizontal autoclave, you have vertical autoclave. Okay. So what is the indicator? The very favorite question, what are the biological indicator which we use in autoclaves? That is Zeobacillus stereothermophilus. Then you have chemical indicator, which is autoclave tape we have, Bowidix tape we have, internal pack control we have, and physical, it will be digital display. Flash sterilization, which use for emergency surgeries, okay, but we cannot use for cannulated instrument. Now we have ETO sterilizer. ETO sterilizer acts by alkylating the cell protein, DNA and RNA. Where we use ETO, we use for heart-lung machine. Please remember, ETO is used for heart-lung machines, sutures, catheters, stents, respirator, dental equipment. What is the control for ETA? So you have a control for your autoclave, you have control for ETA, you have spores of Bacillus atrophius. Okay. In autoclave, you have Geobacillus. Here you have Bacillus atrophius. Now you have plasma sterilization. Here also sterilization control is spores of Bacillus stereothermophilus. We use for scopes like your arthroscope, laparoscope and microvascular instrument. Hot air oven, how it acts? By oxidation mechanism. Controls are spores of Bacillus atrophius. Okay. And what is HEPA filter? Just remember, HEPA filter expansion is high efficiency particulate air filter, which removes 99.97% of particle. HEPA filter, which we use in ICUs, which we use in OTs, and it filters all the particle size less than 0.3 micrometer. Isolation room also, we use HEPA filters. Now, how we sterilize by ionizing radiation, that is called cold sterilization, because we are not increasing the temperature. So we say it is a cold sterilization. Then we have 2% glutaraldehyde for cystoscope and endoscope. We have fumigation of OT by formaldehyde. Peracetic acid is used for endoscope, arthroscope and hemodialysis. So among aldehyde, we have glutaraldehyde, which is 2% for cystoscope, endoscope. Formaldehyde, which is not commonly practiced now, but it was practiced for fumigation of operation theater. Hydrogen peroxide is used for fogging and wound cleaning. Then alcohol, we have sterilium, which we are using commonly for hand rub. Then 70% alcohol for skin antisepsis, and thermometer. Cleaning also, disinfection also, we use alcohol. Chlorine, we have sodium chloride. Sodium chloride, this year question, CT question. What is the disinfectant which we use for blood spill? So we use sodium hypochlorite. We use 0.5% for large blood spill. We use for small blood spill, we use 0.05%. Inspection is used 80 to 85 degrees centigrade, 30 minutes for 3 successive days, which we use for egg-based culture media like Logsdon-Jensen media. Then we have chlorhexidine as a mouthwash we use. Then Savlon, that is 0.3%. What are the tests for chemical disinfectant? So this also has been asked previously. What is the test for chemical disinfectant? So we have Riedel-Walker test, which is also called phenol coefficient test, or Chick-Martin test. Okay. Then we have capacity test, which is called Kelsey-Skies capacity test. Then we have in-use test, that is called Kelsey-Maurer test. So these are the 3 tests which we use for testing the chemical disinfectant. Riedel-Walker test, phenol coefficient test, Chick-Martin test, then capacity test, that is called Kelsey-Skies test, in-use Kelsey-Maurer test. Thank you. So tomorrow we'll discuss some other important points in microbiology.
Best training, peaceful environment, PG medical entrance exam preparation were my expectations. I got them at Speed Medical Village in Kanchipuram. Today, I am a PG topper.
Speed Medical Village, a unique eco-friendly campus dedicated for PG preparation. Speed Medical Institute, a complete solution for medical education.